Okay, so good evening, everyone. It's my great pleasure to be here talking to you about asking a research question. And first of all, a big thank you to the Panacea Steering Committee for inviting me. A little background about myself. I am not an oncologist. I am a pharmacist and a clinical pharmacologist by training. And I have had some experience in oncology. I used to be a research fellow at Trek with Dr. Vikram Gota. So I do have some understanding of oncology. I'm not claiming to be an expert. So what I want to talk about today is sort of give you all an overview of what I believe asking a research question entails, right? Before we begin, here's just a disclaimer slide from Pfizer. So I want to stop here for a second and just let you take this in. When I first got the invite for talking at this workshop, I got really excited because I saw that it was a data analysis workshop, and having been trained in modeling and simulation and quantitative methods used in clinical pharmacology, I thought obviously this would have been an invite for a more quantitative sort of talk. But turns out this is quite a nebulous topic, right? I started thinking about it, and honestly, it felt like the more I thought about it, the more confused I got. And it's going to happen because it is quite a vague topic, and that's what I want folks to understand. It's very open-ended. And this is something that folks develop over the entirety of their careers, and it's more experience-based. And as an early career researcher myself, I want to make sure everyone understands that I don't have all the answers, and I won't claim to have all the answers either. I would ask people to interact, ask as many questions as they can, because we have an erudite group of panelists here and the steering committee as well. So I would ask the panelists and the steering committee to pitch in to answer questions as well, because this is that kind of a talk, and I want this to be as interactive as possible. Okay. So before we get into the nitty-gritties, I wanted to make sure everyone is on board with some of the terminologies that I'll be referring to throughout my presentation, right? So, and I'm not going to give you definitions, but sort of a general overview of what these terms mean so that you can have references to them. So let's start with the problem statement, right? A problem statement is one or many statements that essentially succinctly define your problem. And that could be a research problem, that could be a business problem, whatever it is. As long as it's it describes what the problem is, that's a problem statement. I'm not going to go into details of how we're going to frame that, how we're going to get the most apt problem statement out. But what I will show you is how we come around generating two problem statements. Literature review is something I'm sure everyone must have heard. It's a formal or an informal, but a thorough review of the available literature on any particular topic. Yeah. Then what you have is gap or gaps in knowledge. And this is essentially observations you make while developing problem statements or while performing literature review. You identify the gaps in current understanding of any particular field or topic. And I'll show you how all these three are interrelated, and it's quite interesting. Then there is obviously more study conduct and study design sort of aspects, which is methods, objectives, and endpoints. And I'm sure folks would have heard of this before. And as clinical researchers, I'm sure some of us, or at least all of us, might have heard of this PCOT criteria, which is patient intervention, and so on and so forth. So I'm not going to be describing that because there is ample descriptions of PCOT out there. What I want to really focus on are these four things, like descriptive, exploratory, explanatory, and confirmatory. So these are more about how or what a study is intended to do, or even actually what a research question is aiming to do. Descriptive is essentially just describing a certain condition, patient population, intervention, whatever it is. It's more descriptive in nature, right? Exploratory is something where, let's say there is something unknown about a given condition, and you conduct a study to understand what's happening in it. That's explanatory in nature. Exploratory in nature. Explanatory is something that is further downstream. Let's say you've conducted a descriptive or exploratory study and you've identified correlations and you want to see if there is any physiological understanding to it, if there's any physiological underpinning to it. So you conduct a study that's more explanatory in nature. And the fourth one is confirmatory. Confirmatory is again further downstream from explanatory, which is when you've identified what the problem is, you've sort of figured out plausible reasons for it with explanatory studies, but you conduct another study, right, to confirm your hypothesis. That's a confirmatory study. So I'm going to talk about all of these and sort of show you how they're interlinked. I want to give you some examples of frameworks of how these various terminologies come into play and how they're interlinked with each other. Now, bear in mind, these are some examples. That's why the sum is in bold. Because in my limited experience, I've come across a few different types of research topics and research frameworks. So I'm going to talk about those. And I'm sure there are way more than we can cover in an hour. But this is just to whet your appetite so that you get interested in generating these original research questions for yourself. Okay, so this is framework one, where if you see the flowchart, it's quite simple. There's a problem statement that goes into the literature review, that goes into study design. But what I want you to understand is that it's not always a one-way street. You don't always start with a problem statement. Many a times, you are working in a therapeutic area, disease area, in a particular indication, in a particular drug. And while working in it, while performing regular literature review, you understand that there may be a problem which is unexplored. There is a question that is unexplored, and that is essentially a gap in knowledge, right? So that leads to generation of problem statements. And when you come across these problem statements and write them down, that leads you to conduct further literature review to understand if there is truly a gap in knowledge, or if this required more literature review, or is there something similar that is conducted in an ancillary indication, something that you can borrow from a different indication, from a different drug, right? Many times, physiologically, the underlying pathways are quite similar, and you can make analogies and drive conclusions. And when you have a solid enough understanding of the gap in knowledge is when you get into designing the study. And the study design is something that needs to be well thought out. Some considerations, and again, these are just some, right? Depending on what your problem statement is and what your gap in knowledge is, you need to think about what your study population is going to be. What is the recruitment rate like? What is the sample size that's required for you to be able to understand or get a decent powered study? What are the logistical considerations? Is the study that you're proposing even feasible? And also, of course, last but not the least, ethical considerations, which is honestly should be first and foremost. And this leads me to talk about something called as fit for purpose designs, right? Fit for purpose is actually more a philosophy than it is a framework. And what this means essentially is you design studies or you design trials in a way that answer your particular question, and you're not concerned too much about the additional things that can come with the study design. Of course, in a utopian situation, you can always design a study which will answer 20 different questions. But given the limited resources that were usually dealt with, and the ethical, logistical, and feasibility questions, the purpose of our scientific inquiry should be very clear. And that's where fit for purpose comes into play. You need to design a study that is apt for the question rather than trying to design a study that is, let's say, in general terms, a quote unquote randomized controlled trial. We all know a randomized controlled trial is the gold standard, but sometimes it's just not feasible. It's just not possible. And I'm going to show you several examples of those, well, two in the upcoming slides. So fit for purpose is actually a philosophy rather than just a framework. And fit for purpose can be applied for everything. It doesn't necessarily need to be applied to clinical trials or even clinical research. The field that I work in is called pharmacometrics, where we use modeling methods, quantitative methods to understand how the drug behaves in the human body, pharmacokinetics and pharmacodynamics. I can create a model that is that has, let's say, 30, 40, 50 different parameters. But what's the point when I just need to understand how the clearance changes in a particular population? My parameter of interest is just clearance, right? Or my interest is what is the concentration going to be at this point of time at this dose? That's it. I don't need to really determine all of these parameters. Just an example. So that's what fit for purpose is. And on the other hand, it's something which is way different, but this again goes more towards how we develop research questions. In this, you can see that there are several problem statements, not just one. And this happens when, let's say, you are working in a particular disease area for a very long time, and you realize that on a day-to-day basis, you see these problems in your patient population. You see these questions, you're tackled with them, and you don't have answers to them. And it's not just one question, but several of them. All of them require the same literature review, but then the literature review can generate more problem statements back, and that's why it's a bidirectional flow. And using the literature review, you understand what the gaps in knowledge are. And you can conduct an all-encompassing study which answers all of these questions. But again, you still need to consider the same questions we did before: study population, recruitment, sample size, logistics, and ethical questions. And again, I'm going to throw this fit for purpose in again because I'm a big proponent on it. Yeah, we're developing a huge study design, but then again, there's ways to make it fit for purpose. We can trim off the fat, we can trim off the faff around it, and generate a clean study design. But all of this comes from a solid and rigorous understanding of what your problem statement is. So the key to generating research questions really is understanding the gap in knowledge, doing a thorough literature review, and generating your problem statements. That's where the research question comes from. Now, I'm going to talk about two examples, and these are two case studies that I've worked in, and the third that I've not worked in, but still applies to this conversation because it's something we've not thought about it before, or even if we have, it's not explored as much. The first example I'm going to talk about is a biosimilar analysis a biosimilar of Rituximab called Riditux in diffuse large B-cell lymphoma, right? So this is a study that was conducted, analyzed in ATTRACT. I was a part of it with Dr. Vikram Gota. And this is an example of framework one, where there is a directed question. There's one question to answer. You develop a clean study design for it with all the ethical logistical considerations, and you get to the answer that leads to clinical impact. So let me give you a background on Rituximab, and essentially what I'm doing is showing you what the literature review yields, right? In a usual scenario, I would, if this was like a 3-hour workshop, I would have given you literature and asked you to generate the problem statement. But I'm going to give it to you because we don't have as much time. So Rituximab, some might be familiar with it. For folks who are not, it's a chimeric anti-CD20 monoclonal antibody. And it has revolutionized the treatment of lymphomas. It's the standard of care for several indications, both oncology and non-oncology, actually. Non-oncology, it's used in lupus and in rheumatoid arthritis and several other autoimmune conditions. But the problem is its high cost limits its use in developing nations. Right? Now, the good thing is that there's something called as biosimilars, which are generic equivalents of innovator biologics. But the problem with biosimilars is that their development, production, and approval is way more complex than that for small molecules, which usually requires a BA/BE type study. Riditux is a Rituximab biosimilar produced by Dr. Reddy's, and it's priced at half the price of the innovator. Immediately, you can see that the problem is the cost, right? And there is an alternative available. But what is the problem and what is the gap in knowledge? The problem statement, of course, like we said, Rituximab, the innovator, is expensive and hence difficult to prescribe for patients in developing nations without established economic incentives or established, let's say, healthcare incentives, right? Health insurance. The gap in knowledge is that Rituximab biosimilar is available, but there's a lack of clinical trials demonstrating equivalence of the innovator and the biosimilar. If you took a look at the literature, there is, at that point of time, there was no pharmacokinetic comparison of the two formulations. But what we did have is internal data from TMC, from Tata Memorial Center, showing clinical experience, which showed comparable efficacy and safety. So one of the two things was okay. We had clinical experience of comparable efficacy and safety, but we did not have the pharmacokinetic comparison, which is the other half of the requirement for considering them as equivalent. So a study was undertaken at ATTRACT. It was prospective, open label, single center pharmacokinetic study, right? Prospective means it's being conducted now. We don't have the data. We need to recruit patients. Open label just means that there was no blinding. and it was a single center pharmacokinetic study. Now, for folks who are not familiar with pharmacokinetics, it's essentially the science of understanding how your body deals with the drug. Essentially, it's absorption, distribution, metabolism, and elimination of the drug, right? So let's think about it. If you wanted to do a comparison study, what you would do essentially is it doesn't matter if it's open label or not, but you would have done at least a cohort or a comparator study, right? You would have two arms, one for Reditux, which is the comparator, and then you would have the innovator, right? The reference and the test. But mind you, the biggest thing we're faced with is cost challenge. Cost is a problem. So prescribing, even if you were to develop this quote unquote ideal design, it wouldn't be as feasible because you would not be able to recruit patients unless you had funding from somewhere to essentially buy the innovator product, right? The innovator formulation. So this is essentially, again, quote unquote, a fit for purpose study where you want to understand how PK is similar or different, but you cannot, well, I don't want to say cannot, it's difficult to recruit one arm of the study. So what do? You don't. You just conduct one single arm study where all patients receive the generic. So that's what we did. N is equal to 21, 21 subjects, all received 6 cycles of the R-CHOP with 375 mg per meter square. And that's how the data was generated. We did serial PK sampling in the first cycle up to 21 days. And the concentration titers were measured by an ELISA assay. We also collected B cell counts and survival data. Now, the other question now is, okay, we don't have innovator data from the study. What do we do? How do we overcome this? And the answer is literature review. You look in the literature and you compare it to the literature data. And that's exactly what we did. This is results from our study. On the left, it is the POP-PK model. The top right is the B cell count, where you can see it's completely depleted within the first dose. And on the bottom right is the survival data. It's a Kaplan-Meier curve. Okay, so this is what I was trying to get to, is you look into the literature and you identify what innovator data is available. And that's exactly what we did. Table 3 right here, you can see comparison of the PK parameters of our molecule, which is the generic versus the innovator. And what's interesting is that if you just look at the innovator data itself, it's highly variable. But we still were able to fall within approximations of other studies, especially Blasco and Regazi. And Blasco et al. Was in the same indication as well. And the interesting thing is our model was fairly decent. The numbers or the values within the parentheses are actually your CV %. And you can see that we have a pretty good model. We're able to estimate the parameters with decent precision. Add to that the fact that we have internal data, and this is the publication, which shows comparable efficacy and safety of MabThera, which is the innovator, and Reditux, which is the biosimilar. This gives physicians or the oncologists the confidence to prescribe Reditux instead of MabThera at half the price. And that's an immediate savings for the patient. At the same time, we all know it has revolutionized the treatment. So you're really improving the quality of life of this patient. So this was an example where we started out with one question, a single problem statement. And this is a problem statement, again, that the physicians have noticed, the oncologists have noticed in their clinic. They know that this drug is available. It's probably life-saving for some, but it's just too expensive. And this is, I feel, a brilliant example of how to understand generating research questions. It's also a great example of understanding the considerations you have to think about while designing the study, because sometimes it's just not feasible. Okay, so I'm going to move to the second case study, and this is non-oncology. But I want to show you how framework 2 is used, right? So this is this case study was part of my PhD thesis. So I'm quite familiar with it, but there are still intricacies that I don't understand because it's a huge study, and I'll come to it. The overarching narrative is anti-epileptic drugs in special populations of pregnancy and breastfeeding infants, right? Before we get into it, let me give you a lay of the land. The question is, what's the need to understand exposure in pregnancy and breastfeeding? And by exposure, I mean exposure of anti-epileptic drugs, right? In the mother, there's physiological changes that happen, which usually changes the way the drug is either absorbed, metabolized, distributed, or eliminated. And this leads to changes in concentrations, changes in exposures, and hence, in turn, disease outcomes itself. And this leads to issues such as miscarriages, preterm labor, and so on and so forth. But the issue isn't siloed to the mother. The drug reaches the fetus as well. And this is a huge problem. In-Utero exposure of drugs may lead to teratogenic effects. And sometimes these effects can be dose dependent. Think about a situation where the mother is metabolizing or eliminating the drug faster than non-pregnant state, right? And the physician has increased, the epileptologist has increased doses. Now there is an even higher amount of concentration of the drug or exposure to the fetus, which can lead to further teratogenic effects. And to add insult to injury, the effects don't stop there. There's effects in even infants, because the infants can possibly be exposed via breastfeeding. And breastfeeding, as we all know, is recommended regardless, usually, of up till 6 months, exclusive breastfeeding. But what if the mother is taking anti-epileptic medications? Think about it. It's an anti-epileptic medication. The accumulation in the brain is going to be pretty significant. And it could, in infants, lead to neurocognitive developmental deficits and impaired behavioral development. So the physician, the epileptologists we were working with, and these were from epileptologists from Harvard, Stanford, and also 20 different universities, looked into their own clinics, looked into the literature, and they came up with these problem statements because they had either no answers to these, or there was very limited information about these. For women with epilepsy, it was questions such as, does seizure frequency worsen in women with epilepsy during pregnancy? Do birth outcomes worsen? Right? That's a huge question. Are there more C-sections? Are there more miscarriages? Relationship between the anti-epileptic drug exposure and the outcomes. Does it matter if we increase the dose? Does increasing the dose lead to any altered effects? Or do we even need to alter the dose of anti-epileptic drugs because of pharmacokinetic changes? Do women with epilepsy have an increased risk for depression during pregnancy and postpartum period? Because that's usually a common side effect of some of these anti-epileptic medications. And the next question is for the children born to women with epilepsy, right? That's the bigger category. What are the long-term effects of in-utero anti-epileptic drug exposure on verbal intellectual abilities and other neurobehavioral outcomes? How do neonatal outcomes differ? And by differ, I mean differ when compared to children born to healthy pregnant women, right? Does breastfeeding, when taking anti-epileptic drug, impair the child's ultimate verbal and other cognitive outcomes? Now, each of these questions is standalone. There can be a single trial for each of these questions. And mind you, there are small studies that have been done to answer these questions, but that's it. They're small. So what are the gaps in knowledge? There are registry studies that show that some anti-epileptic drugs have lead to major congenital malformations, are probably teratogenic as well. But they are registry studies. They don't give you information that they've not collected. There's no information sometimes on dose. There's no information sometimes on exposure. Usually there's no exposure information. So there's a huge drawback with registry studies, although highly informative, but there's still gaps in knowledge. The other prospective studies that have been done are small clinical observational studies, which do not have any exposure response characterization. The other interesting part that we found out was that there was no follow-up done in postpartum for many of these women, or in many of these studies. They just looked at the pregnancy, and that's it. What happens post-pregnancy? No one knows because no one's done it. And there was no comparator groups either. And the comparator groups usually depends on the question, but they can either be healthy pregnant women, or they can be non-pregnant women with epilepsy, depending on what the question is, and we'll get to that. There's also issues with consensus. US has different way of treating pregnant women with epilepsy to the EU. And that's a huge debate. Some of them recommend therapeutic drug monitoring in the US. And in the EU, they say therapeutic drug monitoring is really not necessary because we don't think changes in concentrations are actually leading to increased seizures. There's also a debate between epileptologists and pediatricians versus OBGYNs on breastfeeding. The epileptologists and pediatricians are proponents of breastfeeding even while on anti-epileptic drug because registry studies have shown no untoward effects. But OBGYNs are more conservative and say, pump and dump. You don't need to give milk to the child when you're taking anti-epileptic drug. And the issue is, throughout this entire duration, pregnancy and postpartum, mothers cannot stop anti-epileptic drug because that could impair quality of life and sometimes even survival. Using all this literature, what we sort of concluded was that a lot of this comes to issues with study design and working with this population, with the special population itself. think about it. If you wanted to answer all of these questions, you would need a really long enrollment period to just reach to the adequate sample size. There would be recruitment issues. There would be issues regarding screening, rescreening, and in that you would have missed the window. There would be ethical considerations on PK sampling. How much blood can you collect from these individuals? And if you, for example, if you wanted to measure concentrations or exposure in infants, you would have to poke the infants and think about, I mean, which mother would be okay with that when you're collecting routine samples from their 2-month infant and routinely sticking them? There's also this problem of infant measures, neurocognitive, behavioral development, which can only be collected, which can only be measured after a long time, because it needs either at 2 years or at 6 years is usually when these measurements are done. So you would essentially for 2 to 6 years, sorry, not 2 to 6 months, sorry, apologies, 2 to 6 years before these outcomes are available to you. So there's huge questions being asked here. And to tackle this, before we go to that, this is just a slide on the different physiologic and drug disposition changes that happen in pregnancy. You can take a look at this afterwards, but essentially, due to this, clearance usually increases, which means that the drug is being eliminated at a faster rate than non-pregnant, which means you won't have therapeutic concentrations in the system. So you would probably have to increase doses. Sorry, but I was talking about this, right? So the study that was designed to answer these heavy questions was MONED. It's maternal outcomes and neurodevelopmental effects of anti-epileptic drugs. It's a NIH-funded 20-site prospective observational parallel group cohort study. And it recruited 3 different groups. Pregnant women with epilepsy, we ended up with 351. Children born to pregnant women with epilepsy, 345. Non-pregnant women with epilepsy, which was 109, and healthy pregnant women, 105. Now we have 2 comparator groups, like I said, and we will be using them to compare with the pregnant women with epilepsy, depending on the question, right? And the objectives of the study is here. Is there a worsening of seizures during pregnancy? And what are the contributing factors? Is there an increase in C-section rate in women with epilepsy? And what are the factors? Is there an increase in depression, right? For children, what are the long-term effects of in-utero anti-epileptic exposure? Is anti-epileptic exposure, anti-epileptic drug exposure correlated with small for gestational age outcomes? And if breastfeeding while taking anti-epileptic drugs, does that impair the child's ultimate verbal and other cognitive outcomes? So we designed a study to answer these specific questions, but we still had to compromise on some things, and that is in the study design. On the left is some of the inclusion and exclusion criteria, but on the right, you have the study protocol. And I just want you to look at this protocol and see how long the study is. And this is per individual. It took us 6 years to recruit all of these patients to reach our sample size as calculated by the power analysis. For pregnant women with epilepsy, we followed them throughout pregnancy and 9 months into postpartum. And for non-pregnant women, it was a total of 18 months. And we had about 7 visits, 4 during pregnancy, 3 during postpartum, and just 7 in general for non-pregnant. And these stars you see, these asterisks that I've put on, those are blood draws that we did. But it's not intensive blood draws, it's single blood draws. And if you've ever done a pharmacokinetic study, you'll realize that single blood draws are usually way too less of a data, right? If you've done BAB studies, you will see there's approximately 12 to 15 blood draws to characterize the entire PK profile, right? But we can't do that because if we wanted to do that, we would have to ask pregnant women or even non-pregnant women with epilepsy to come in at 4 times or 7 times, depending on which group you're in, keep them overnight and draw excessive blood. Well, it's ethically would be, physiologically, sorry, would be still okay, but ethically, not so sure. And then you also need blood draws from infants. Our initial proposal was to collect 2 samples, one 2 weeks after birth, and the second one was about 12 weeks. But think about the logistic and feasibility, right, concerns here. Asking women who've just given birth to come back at 2 weeks just so that we could get their infant's blood is going to be a straight no. So we ended up with just a single blood draw for the infant. And this is the data we have in terms of drug exposures. This is it. I'm not going to show you where I contributed to the analysis because that's not relevant to the study. But I just want you to understand that all of these were great questions which need answers, which I mean, the physician, the epileptologists need answers for this because it's clinically relevant. And honestly, the results from the study are practice changing. But we still couldn't do an ideal study because it was just not possible. Even though you've generated the apt research question after doing a thorough literature review, it was just not possible. Okay, the last case study is something that's different, and I want to pivot to it because we don't really see research like this often. And this is a very unique sort of study that I want to show you. It was developed by Bhumika Desai, who was also a research fellow at TMH and at TREC, at Credo, and it was presented at Asian Health Literacy Association. So courtesy Bhumika Desai, thank you. The overarching narrative is understanding information-seeking needs in oncology patients. The past 2 examples I gave you were very clinical research heavy, very drug heavy, very clinical trial heavy. But this one is different, but it still is relevant because it's about how to identify a research question, how to identify a problem, sorry, and generate a research question, right? So just to give you a background, in India, oral cancer is the 3rd most common type of cancer and accounts for about 30% of all types of cancers. And honestly, it's a very complex disease, as of course, oncologists here might be aware. There's the disease, there's the treatment, there's the associated psychosocial impact, and all of this leads the patients and their family members to encounter both information needs and emotional support needs throughout the course of the disease. Now, I'm not a like clinician. I'm not a diagnostician either. So I don't really know. But as someone who's in the field looking at these patients every day, this question came up, which is there are particular needs pressing in these patients and their family members' experience. Um, Significant decrements in quality of life, obviously. There's high levels of emotional stress, and there's deterioration in interpersonal relations and increased social isolation, of course, because it's such a terrible disease. Now, if you could present the right information to these patients and family members, it would not only help them with decision making, but it also prepares them for the treatment, helps them understand and cope with the adverse effects, reduces anxiety and depression, increases satisfaction with treatment, improves communication between not just with the family, but also the healthcare providers, and hence overall improves the quality of life. And at the end of the day, apart from treating patients, uh, quality of life improvement is also something that I think oncologists and physicians strive towards generally. But these type of questions is not something I've thought about a lot. So I really wanted to talk about this because it's so unique, but it's so necessary. These kinds of questions need to be asked. And folks who are in these situations are honestly can look around and identify these questions, even if it's not in their research area of interest, even if it's not in their expertise. Okay, so what are the problem statements? What are the information needs of newly diagnosed OSSC patients and family? What is the information seeking behavior like in this population? And which areas need improvement for healthcare professionals? These are pretty clear and succinct problem statements. The question isn't to provide them with information, it's a step behind, it's a step upstream, which is to just understand what the information needs are. And once those are understood, you can move towards deploying or generating frameworks of how to disseminate that information. So this is the framework that was developed, which is essentially you have OSSE patients about 18 years of age with 1 month of new cancer diagnosis and physically and cognitively able will be considered eligible. Of course, you will need informed consent. Face-to-face in-depth interviews to be conducted, approximately 25 patients to reach data saturation. You will have to do interviews in Hindi, English, and it could be about 45 minutes and audio recorded. An interview guide will be used to assist the field investigator cover relevant topics. Patient and caretaker information, experience with healthcare provider, prior knowledge about cancer, cancer information seeking, and health information needs. These are the topics that need to be covered. And how do you analyze it? Interviews will have to be transcribed, right? You will have to generate transcripts of interviews conducted in both Hindi and English. Hindi needs to be converted to English. And three-stage coding. Initial coding, which is intense line-by-line analysis of transcripts. Focused coding, which is selected set of most prevalent codes will be identified. And theoretical coding, final categories in resulting theory, right? So what all this generates is a theoretical framework. That's the final outcome, right? And this essentially will account adequately for all data gathered while seeking to explain the process of cancer information seeking and information needs, and ultimately leading to patient satisfaction with hospital and or healthcare provider. this question was this problem was identified when Bhumika was working as a research fellow in this DMG, where she realized that a lot of folks, a lot of patients did not necessarily understand, owing to a whole bunch of different things, the different treatment modalities, the different options, the adverse effects. And all of this led to a drop in quality of life. So my wrapping up statement would essentially be that there's different problems out there, and it's, I think, up to us to identify what the gap in knowledge is, what is not available, and generate apt research question based on rigorous literature review and rigorous understanding of the field or domain itself. And this doesn't necessarily have to be limited to clinical research. This is true for every research that is done. You really need to have a good understanding of the lay of the land. You need to understand what is and what isn't available, leading to the gap in knowledge, and what needs to be answered, which ends up becoming your research question. So if you were to ask me what the recipe for success is, I don't think I have an answer, because it's not a one-way, two-way street. It's a complex highway interwoven with so many different things. And the frameworks that I've shown are the ones that I've encountered. I'm sure there are way more experienced folks here who have come across various different situations of how they've generated research questions. Sometimes it's spontaneity, sometimes it's more of an expedition to look for a question. Sometimes it's just clinical experience. And with that, I come to the end of my presentation. Once again, thank you for listening to this really vague presentation. I hope there was more structure to it, like the other data analysis presentations, but I hope you got something fruitful out of this. And a huge thank you to Panacea for inviting me. I am very early in my career, and I was very, very excited, but at the same time terrified of presenting here. So I hope this was an apt appetizer to whet your appetites for the upcoming plethora of presentations your way. Thank you.